I'd like to welcome everybody and thank you for joining our webinar today. My name is Kevin Babachek. I'm the Director of Product Management for QuantaBio. And as a quick reminder, this webinar is being recorded. But with that, I'd like to move ahead and introduce our speaker for today, Dr. Subrata Panja. Subrata has been with QuantaBio for more than 3 years. He's the Associate Director for Field Applications for our team. He has more than 20 years of research experience in the molecular biology area. Previously, before he came to Quanta, he was involved in diagnostic assay development with different tools such as NGS, qPCR, and digital PCR while at GeneDx. He has a very exciting talk today to really look at some of the improvements in PCR that have direct applicability to next-gen sequencing applications. I hope it's very interesting for all of you. A couple housekeeping items during the talk. If you would please keep your microphones muted to avoid any background noise so everybody can hear. If you do have questions, please use the Q&A button within Zoom. And then at the end of Subrata's talk, we'll go ahead and go through any of those questions that you may have. And with that, I will turn it over to you, Subrata.
All right. Thank you, Kevin. First of all, thank you everybody for joining this exciting webinar from QuantaBio. So today we are going to talk about next generation PCR amplification technologies and how that is improving different next generation sequencing applications. Now, PCR or polymerase chain reaction is a household name for almost all molecular biology or genomic labs. But that all started almost 70 years back with the innovation of DNA double helix. First, fast forward in 1980s, when PCR or polymerase chain reaction first developed, was really exciting for the field of molecular biology. Later in that decade, the development of Taq DNA polymerase and first automated PCR cycler took the field to the next level and helped adopting this technology in different molecular biology laboratories. Further development happened in 1990s, where proofreading polymerases were developed. Antibody hot start gave capability to set up the reaction in room temperature stability. It helps leaky amplification and different advancement. Around 2000, different biotechnology companies combined all the development happened so far and took the field to the next level, developed different advanced amplification enzymes or master mixes, which really improved different applications like molecular cloning, Sanger sequencing. Even it was really helpful for early development of different kind of next generation sequencing technologies. But the last major development on the amplification technology we remembered was Q5 from NAB, which had significant impact on different NGS sequencing applications. Now, what happened between 2012 and 2023? Last 10 years, we have not seen any major development on the amplification technology. But other technologies and applications developed in astronomical fashion. That includes digital PCR, second and third generation sequencing technologies, including whole genome sequencing, RNA sequencing, single cell sequencing. Even we are seeing huge development on the spatial transcriptomic applications. But again, we have not seen major development on the amplification side. Still, demand is the same. We all want high fidelity enzyme or master mix with faster amplification rate, long range capability, inhibitor tolerance capability, so that if samples are coming from water, soil, fecal samples, or other kind of environmental sample which inherently contain higher amounts of inhibitor, the PCR reaction should be able to handle that. It should be able to overcome high GC or high AT rich biases. And if possible, whether these enzymes or PCR master mixes can amplify directly from sample or without going through really lengthy extraction processes. Now, most of the enzymes or master mixes available in the market is performing well and can fulfill some of the features or attributes I mentioned here, but not all. So in 2020, we decided to develop a solution which can address all the attributes I mentioned here, if not all, at least most of the features I mentioned in this particular slide. So we developed a product, but we were not able to bring more information in front of you because of COVID. So post-COVID, we want to share more information about our next generation amplification technology in front of you. And we are really happy to talk about repliQa HiFi ToughMix and share some application information with you. So repliQa HiFi ToughMix is a fast polymerase with extension speed as fast as 1 kb per second, long range capability up to 40 kb, and it is a high fidelity PCR master mix with 90x fidelity compared to wild type tag. We shared the reagent, or this reagent was tested by different labs, and we got overwhelming amount of positive feedbacks. And you can see some of the feedback I included in this slide definitely improve the performance and the data quality they received using repliQa HiFi ToughMix. So in next couple of slides, we'll talk about some features and benefits of repliQa HiFi ToughMix. So as I mentioned, repliQa HiFi ToughMix is a high fidelity master mix. Fidelity is 90 times higher than wild type tag. It is a super fast PCR master mix or polymerase. The speed or extension rate is at least 3 times faster than most of the polymerase available in the market, with extension rate as fast as 1 kb per second for less than 1 kb targets. It is a ToughMix product, so it has increased or higher tolerance to a wide range of PCR inhibitors. It is also a highly sensitive reagent, and it can amplify targets with inputs as low as 2 picogram. And finally, it has long range capability. It can amplify up to 20 kb of genomic DNA targets and up to 40 kb of lambda DNA or other DNA virus targets. So let's look into few data to support all the features I mentioned here. So in this particular set of experiment, we took E. coli genomic DNA as a template and amplified targets of different lengths. As you can see at the lower end, we were able to amplify targets around 200 to 250 base pair length, all the way up to 20 kb. And at different lengths, you can see repliQa generates really nice looking band. And it is specifically exciting to see a really nice looking band at longer targets, because typically long range PCR gives you specific bands, and smear. So being able to see really nice looking band at 20 kb target was really exciting. As I mentioned, speed is a really important feature of repliQa HiFi ToughMix. It can, the extension rate is between 1 to 10 seconds per kb, depending on the target length. Just to give you an example, here we amplified a 2 kb fragment using repliQa HiFi ToughMix or some of the other products we tested here using manufacturer's recommendation. And repliQa took only 23 minutes to amplify 2 kb target, which was 50% less time took by other polymerases we tested in this particular experiment. Now, if you use even longer targets like 10 kb, 15 kb, or 20 kb, the time taken by repliQa to amplify those targets and time taken by other polymerases, you will see a greater difference. Long time back, long-range PCR, people used to do only overnight because long-range PCR usually take 6 to 8 hours. When people are using repliQa HiFi ToughMix, no longer overnight PCR reactions are required, and you can amplify 10, 15 kb targets in 2 to 2.5 hours. repliQa HiFi ToughMix is also a highly sensitive product with, and it can give higher yield. Again, in this particular experiment, we amplified gDNA templates of different length, different GC content, and the average yield we observed with repliQa HiFi ToughMix was higher than some of the other reagents we tested in this particular experiment. And at the end, we want to talk about inhibitor tolerance capabilities of repliQa HiFi ToughMix. Inhibitors are present in many samples in different forms. For example, hematin is present in blood sample, xylin present in plant tissue, humic acid present in the soil sample can inhibit PCR reactions. So PCR reagents having the capability to tolerate higher amounts of PCR inhibitors are extremely critical to amplify DNA targets from those samples. And you can see here that repliQa HiFi ToughMix has higher tolerance to the inhibitors I mentioned here compared to some of the enzyme we tested. Not only these 3 inhibitors, we have tested many different kind of inhibitors, and repliQa HiFi ToughMix were able to withstand higher amounts of inhibitor in the PCR reaction. Now, as you can imagine, everything we tested in the lab was started with pure DNA and then adding different amounts of inhibitor, and you may be thinking that may not be the real life situation. And we learn more about the capability of our reagents from different labs, testing our reagents at the same time, different publications coming in the peer-reviewed journal, and we are learning more about capability of our reagents from there. So I want to share one publication with you, which was published in 2022, where they were trying to amplify targets from an insect without going through DNA purification. So they crushed different parts of the insect and then extracted the nucleic acid using TE buffer. So this is basically a crude extraction. And they tested repliQa HiFi ToughMix and other inhibitor-tolerant PCR amplification reagents available in the market. And this is the data looks like. You can immediately see that repliQa has thickest band compared to other polymerases showing here, indicating that repliQa gives higher yield even in presence of inhibitors. Because as you can imagine, crude extraction like this have a lot of inhibitor and other substances present in the reaction. Even if you look at the statistics presented by that publication, repliQa HiFi ToughMix was able to amplify 27 samples out of 28. And the next best product were able to amplify 14 targets out of 28. So definitely this suggests or justify our claim that repliQa HiFi ToughMix is a true ToughMix product, can withstand inhibitor present in the samples. Keeping all the features in mind, we wanted to see how repliQa HiFi ToughMix can improve different applications for next generation sequencing. In rest of the presentation, I'm going to focus on Amplicon-seq application, emphasizing on 16S metagenomic sequencing. Then I'll talk about rapid RNA virus sequencing. And at the end, I will emphasize on whole genome library amplification. Let's start with Amplicon-seq, emphasizing on 16S metagenomic sequencing. 16S metagenomic sequencing in Illumina platform, most of the lab amplify 460 base pair target between V3 and V4 regions using locus-specific sequence flanked by adapter sequence. After the first round of PCR, a bit cleanup to remove free adapters or other components, and a second round of PCR to incorporate Illumina Nextera adapters. A final bit cleanup, and then pool all the samples and sequence it in MiSeq or other sequencing platforms. Now, this kind of workflow is not only applicable for 16S metagenomics, rather it can be applicable for any other kind of Amplicon-seq workflows. Historically, people used Kappa, HiFi, or other polymerases. In our hand, Spark HiFi PCR master mix from QuantaBio showed equivalent performance compared to Kappa HiFi. So in rest of the presentation, we are going to compare the performance of repliQa HiFi ToughMix with Spark HiFi. When you use Spark HiFi or Kappa HiFi for 16S application, this entire workflow takes approximately 3 hours. Replacing Spark HiFi with repliQa HiFi ToughMix reduced the overall workflow from 3 hours to 2 hours. Now we can remove the bead cleanup between two different steps, and we can perform the entire workflow within 1.5 hours. In our lab, we mixed 3 different bacterial species with different or wide GC content and wanted to see if repliQa HiFi ToughMix can identify those 3 bacterial species in regular workflow and the workflow where we removed the bit cleanup step. As you can see that both Spark HiFi and repliQa HiFi were able to identify those 3 bacterial species. Even the result was same when we used repliQa HiFi ToughMix without the bit cleanup between 2 different PCR steps. As you can imagine, this is a very simple experiment where we mixed 3 bacterial species. But if you want to identify bacterial or microbial population present in soil, water, or fecal sample, there may be 20 to 30 or even more bacterial species present in the sample. To understand how repliQa is working in those kind of situations, we will refer a recent publication published in this year. In this particular case, they were interested to understand fecal microbiota composition in black-tailed prairie dog using 16S metagenomics. And in this particular sample, they were able to identify 11 phyla, 57 families, 140 genera, and 220 species present in the fecal sample. As you can imagine, in order to understand 220 species, and even some of the species present in very low copy number, the amplification enzyme have to be highly sensitive, at the same time should be able to overcome higher amounts of inhibitors present in the fecal sample. Now, repliQa HiFi ToughMix is not only beneficial or improving the Illumina-based short read workflow, it is also helpful improving 16S metagenomic workflow in ONT platform as well. So someone can purify DNA from soil, water, or other sample, amplify the entire 1.5 kb region of 16S gDNA, and then prepare library using ONT barcoding kit, and then sequence it in nanopore or Oxford nanopore or different platforms. Now, if you replace the third-party recommended PCR enzyme or master mix with repliQa HiFi ToughMix, you can immediately save a lot of time. At the same time, the inhibitor tolerance capability of repliQa HiFi ToughMix is improving the detection capability and success rate of 16S metagenomic sequencing in different sample types. Just to give you one example, one of our collaborators isolated DNA from soil samples and water samples and used repliQa HiFi ToughMix and observed really good quality data. In this particular example, some of the bacterial species identified is mentioned on the left-hand side, and the taxonomic tree was generated with higher confidence. So overall, repliQa HiFi ToughMix is a good choice for most of the Amplicon-seq applications, including 16S metagenomic sequencing in Illumina or long-read sequencing platforms. Now I want to move to the next section of this talk, where I want to talk about rapid RNA virus sequencing application. This project started during COVID because that time different labs were trying to develop sequencing workflow to sequence coronavirus RNA. Most of the labs used Arctic-seq or Amplicon-seq based protocol where a high multiplex PCR primers were used, where it include 30 or higher plex PCR primers were included in the reaction. To develop that kind of high multiplex PCR primer set required a lot of work, a lot of optimization. Now, how people can adopt that kind of workflow in multiple different RNA virus sequencing? Rather than going through that extensive primer design process, we took a different route, which is much simpler and has some added advantage. In this workflow, we started with isolated viral RNA and then generated longer cDNA with gene-specific primers just in 10 minutes using one of our innovative cDNA synthesis kit called QScript Ultra Flex Kit. Once we generated long cDNA products, then we used repliQa HiFi ToughMix to generate long PCR products just within 90 minutes. Once we have that long PCR products, you can use it to sequence it any sequencing platform. If you want to sequence it in short read platforms, you can fragment it, generate libraries, and sequence it. At the same time, long PCR products can be sequenced in Oxford Nanopore or PacBio platform as well. In next few slides, I'll show you the data generated for Illumina sequencing, where we used Spark DNA Frag and Library Prep Kit to generate library for Illumina platform and sequence it in MiSeq. So just to give you a little more understanding what we did here, we started with isolated coronavirus RNA, which is approximately 30 kb long. We used a pool of 4 gene-specific primers, which binds at 4 different regions of the coronavirus RNA. We generated long cDNA using QScript Ultra Flex Kit, and then we generated approximately 8 kb length of amplicons using repliQa HiFi ToughMix. So this is how individual targets look like, and you can see all the targets are really nice looking and clean. As I mentioned, once you have this longer PCR products, you can sequence it in any platform. What we did, we fragmented the DNA and prepared library for Illumina platform. So these are the libraries generated for Illumina platform. Once we sequenced it, we looked into some of the sequencing matrices, and you can see that percentage of map reads were between 97 to 99%, and even with Amplicon generators, long Amplicons generated using repliQa HiFi ToughMix, the duplication rates were really low. When we used those reads and mapped into coronavirus genome, we were really happy to see that we observed uniform coverage throughout the genome. And in some regions, you can see the amount of reads are double, and that's coming from overlapping regions from different long-range PCR products. And these overlapping regions are really helpful to detect the mutation present where primers are binding. Another advantage of this long range based application is that obviously it can detect all the single nucleotide variation present in the RNA virus. At the same time, if there are structural variants present in the RNA sequence, long-range PCR products will be able to identify those structural variants like deletions or duplications really easily or more efficiently compared to shorter amplicon-based approach. Now, coronavirus RNA is 30 kb long, but many other common RNA virus like HIV, HCV, poliovirus, those RNA viruses are less than 10,000 nucleotide long. And different labs are utilizing this method by just using one gene-specific primer at the 3' end to generate the entire cDNA in 10 minutes, followed by long-range PCR and sequencing the entire RNA virus. So this method is more versatile and definitely helping understanding different kind of mutations present in RNA viruses. And at the last part, I want to talk about whole genome library amplification and advantages using repliQa HiFi ToughMix. So whole genome library preparation process involves different steps. First step is fragmenting the DNA either by mechanical or enzymatic process, end polishing, followed by adapter ligation, a bead cleanup to remove free adapters, and library amplification to get enough library for sequencing, and finally a bead cleanup. Throughout the process, we are going to focus on the library amplification step only. Few years back, when we launched Spark HiFi PCR master mix, we observed better performance, higher yield from Spark HiFi PCR master mix compared to some well-known master mix present in the market. So in rest of the presentation, we will only compare the performance of repliQa HiFi ToughMix with Spark HiFi PCR master mix. To understand how repliQa HiFi ToughMix amplify whole genome libraries, we mixed 3 different bacterial species in 1:1:1 ratio, and these bacterial species have GC content of 27%, for E. coli 50%, and for Bordetella pertussis 67%. Mixture of 3 bacterial species will give us a better understanding of a target with wide range of GC content. In this particular example, we generated library with 100 nanogram of mixed bacterial genomic DNA target, and the libraries were amplified either with Spark HiFi PCR master mix or repliQa HiFi ToughMix with either 1 second extension time or 30-second extension time. And in each case, we observed equivalent yield, suggesting that repliQa HiFi ToughMix will produce enough yield just within 1 second of extension time. We also used lower input, like 10 nanogram of mixed bacterial genomic DNA target with 9 cycles of PCR. Even with 1 second or 30-second extension time, repliQa HiFi ToughMix gives almost comparable yield with Spark HiFi PCR master mix. We also looked into the duplication rate generated by Spark HiFi PCR master mix and repliQa HiFi ToughMix. And you can see that in both 100 nanogram input or 10 nanogram input, duplication rates were lower, either with Spark HiFi PCR master mix or repliQa HiFi ToughMix, irrespective of 4 cycles of PCR or 9 cycles of PCR, even when we did the extension of 1 second with repliQa or 30 seconds of repliQa. Overall, our data suggest that repliQa HiFi ToughMix is a good candidate for amplify whole genome libraries. We also looked into the coverage across a wide range of GC content. And you can see that from 15 to 85% GC range, Spark HiFi, repliQa HiFi with 1 or 30-second extension showed exactly same kind of GC coverage across the genome, showing that both Spark HiFi and repliQa HiFi ToughMix have minimum bias towards different GC content. And which is important because different laboratories are sequencing human genomic DNA, bacterial genomic DNA, and all different kinds of genomic DNA. So having minimum bias during PCR amplification is extremely important. Now, I mentioned before, whatever we are doing in our lab is sometimes limited to the kind of samples we are using. And we are learning more from different publications coming in peer-reviewed journal. So at the end, I want to share a recent study published in bioRxiv. The article titled Identifying the Best PCR Enzyme for Library Amplification in NGS. This article submitted by Wellcome Sanger Institute. In this particular article, they evaluated 20 different high-fidelity PCR enzyme or NGS amplification mixes. They used 4 different bacterial targets with 67.7% GC content, 50% GC content, 29.1% GC content, and 19.3% GC content to look into amplification bias based on the GC content of the target on all 20 different high fidelity master mixes they have tested in this study. Their target insert size was 500 base pair, and they used 1 nanogram of PCR-free library fragments from each genome, and they amplified using manufacturer's recommended conditions. Let's first look into the yield coming from all different targets. If you look into the yield coming from repliQa HiFi ToughMix with different GC containing genome, you can see that repliQa HiFi ToughMix gives higher yield compared to other HiFi PCR master mixes tested here. Not only that, in some of the master mixes, you may observe higher yield with E. coli, which has kind of neutral GC content. But when you go to high GC targets, yield goes down for other PCR master mixes, where repliQa gives high yield for all 4 genome tested in this particular experiment, suggesting repliQa has higher yield and unbiased higher yield for different genome targets tested in this particular experiment. This particular data is really important, where they looked into low coverage index. For their whole genome sequencing experiment, they sequenced the genome with a target of 30x coverage. And they looked into different regions of the genomic coverage and checked which regions has less than 15x coverage, and they calculated low coverage index. And at the end, the sum of low coverage values compared with coverage obtained from PCR-free libraries. And as you can see, this is where PCR-free library resides, and repliQa HiFi ToughMix has low coverage index very much comparable to PCR-free libraries, suggesting that repliQa HiFi ToughMix have minimal bias at each giving coverage uniformity similar to that seen in PCR-free libraries, which is really important information, suggesting that repliQa has minimum bias and results are comparable to PCR-free libraries. Not only for Illumina-based whole genome amplification, they studied the effect of different high-fidelity polymerase in long-read sequencing applications as well. So for that particular set of experiment, they used shared Saccharomyces cerevisiae DNA and size fractionate using either Sage Sciences ELF or Blue Pippin instrument. And the size fraction was either 21.6 kb or 13.3 kb. And the input amount they used for PCR amplification was 1 nanogram. And finally, the amplified libraries was sequenced in Pacific Biosciences HiFi sequencing platform. Now, all the high fidelity master mixes they tested, most of the enzymes did not amplify long range PCR products. So they eventually end up testing only handful of PCR master mixes. Here we are going to show only data from repliQa HiFi ToughMix. So HiFi subread length distribution for ELF or Blue Pippin fractionation of repliQa amplified fragments were tested after 15 cycles of PCR. Now, as you can imagine, long-range PCR can often preferentially amplify smaller templates such that after multiple cycles of amplification reaction, the reaction can be dominated by only shorter amplicons. When they looked into ELF size fractionated DNA, the majority of the reads were obtained from shorter amplification product, and repliQa gave the largest fraction of 20 kb subreads. For Blue Pippin, on the other hand, the average subread length was 12 kb. But overall, even for long read sequencing application, they observed repliQa HiFi ToughMix gave better performance. Finally, they also looked into the fidelity of repliQa HiFi ToughMix and different master mixes. This is also another important factor, and they observed that repliQa does not have compromised performance on the scale of fidelity as well. So when looked into single nucleotide or mismatch frequency, repliQa HiFi ToughMix has equivalent performance compared to all the reagents tested here. Even for insertion or deletion rates were kind of comparable to the really high fidelity master mixes included in this particular test. Now, if you consider the fidelity, the ability to amplify wide range of GC target in unbiased fashion, ability to amplify whole genome libraries for short read platform and long read platform, overall, repliQa HiFi ToughMix performed really well in all different tests. So we can definitely consider repliQa HiFi a versatile amplification technology or master mix for different next generation sequencing applications. So what we discussed today, compared to similar kits, repliQa HiFi ToughMix demonstrate extreme speed, which will reduce the PCR runtime more than half. It has superior sensitivity. You can get higher yield compared to other polymerases tested, with strong inhibitor tolerance capability for wide range of inhibitors present in different samples. repliQa HiFi ToughMix is ideal for 16S or other Amplicon-seq applications for rapid preparation of libraries for both Illumina or other short-read platform, as well as long-read sequencing platforms. Combination of long cDNA synthesis by Q-script ultraflex kit followed by long-range PCR using repliQa HiFi ToughMix is ideal for rapid RNA virus sequencing. This method can detect multiple single nucleotide variation as well as structural variation like larger deletion present in the sample. And finally, whole genome library amplification with repliQa HiFi ToughMix saves time without affecting the yield, duplication rate, genomic coverage, and other sequencing matrices we are interested in our lab. Finally, I want to thank you for attending and listening through this presentation. And I will be happy to answer if you have any question. And please submit your question to the Q&A tab.
That's great. Thank you very much, Subrata. Yes, anybody has any questions, please, you do use the Q&A button in Zoom. We do have a couple already. I can start going through those while other people are thinking of any potential questions. Subrata, the first one is, can you talk a bit more on how repliQa works with high GC targets?
We have tested some high GC targets in our lab. I do not remember all the names, but some GC targets which were not being able to amplify by wild-type tag and required DMSO or other kind of additives to amplify those targets, repliQa was able to amplify without any support. Now, that being said, there are some targets with GC content above 78 or 80% may require addition of some DMSO. But other than that, we observe repliQa can amplify high AT regions, high GC regions without any problem.
Okay, another question is, have people used repliQa for Sanger sequencing in cloning applications?
Yes, absolutely. As I mentioned, in this particular slide, we focused on next generation sequencing applications only, but different labs used it for cloning application. And as you can imagine, generating really clean amplicons are really important for cloning applications. So different labs are using it for cloning applications, getting good results. And I heard feedback like they use other master mixes and were not able to get the right target. When they used repliQa, they were able to get the right target. They had successful cloning. Same thing for Sanger. Many labs, when they use repliQa for Sanger sequencing, they start getting clean sequencing results. So the answer is yes, people used it for both cloning and Sanger sequencing applications got really improved result from there.
Next question is, have you tested this repliQa HiFi ToughMix in wastewater samples to sequence SARS-CoV-2 genome by shorter amplicons such as 400 to 600 base pairs?
So different lab tested this with shorter amplicons, but as you can imagine that like shorter amplicon-based panels required multiplexing with repliQa HiFi ToughMix or other polymerases. We are currently working on the multiplexing capability of repliQa HiFi ToughMix, but if it is not multiplexing, if you're just looking into a few targets, repliQa worked really well with wastewater samples as well.
What is the shortest sequence that can reliably amplify, i.e. less than 100 base pairs?
At this time, I do not remember if somebody amplified less than 100 base pair targets. But again, if you are interested, please try it and let us know how it works. We always learn the capability of repliQa from the development or tests done by different labs. So I do not remember at this time, but yeah, please feel free to try it and see if it works.
Can you talk a little more about the UltraFlex kit that you mentioned during RNA virus sequencing?
Absolutely. So last year we developed and launched QScript UltraFlex kit, which has capability to include flexible priming. You can use gene-specific priming, oligo-DT priming, or random primers for your cDNA synthesis need. What is the advantage or the benefit of QScript UltraFlex kit is that it can generate long cDNA like 20 kb just in 10 minutes. And at the same time, cDNA synthesis is done at 55 degree that helps RT to go through RNA sequence have secondary structures. Even if you need to go beyond 20 kb, if your RNA is intact, you can generate even longer cDNA by performing RT reaction more than 10 minutes. At the same time, UltraFlex kit also have higher inhibitor tolerance capability. So if your samples have inhibitors, UltraFlex kit can also withstand different kind of inhibitors present in the sample. So a combination of UltraFlex kit and repliQa HiFi ToughMix are really beneficial for sequencing or other applications from the RNA targets.
And maybe this is a related question, but the question was, would you recommend a better mix for cDNA synthesis from RNA samples? I know you're just talking about the UltraFlex kit.
Exactly. I mean, it all depends on your end goal. If you need gene-specific priming or oligo-DT priming for your RNA targets, UltraFlex kit is a good solution for cloning or for sequencing application. Now, if you are doing qPCR after cDNA synthesis, we have a master mix version of QScript UltraFlex kit called QScript Ultra SuperMix. So if you are just looking into cDNA synthesis using random primers and end goal is qPCR or short endpoint PCR, QScript Ultra SuperMix is the way to go. For all other applications, QScript UltraFlex kit will be a better solution.
One just came in. Has the product been tested for SNP detection?
QScript UltraFlex or repliQa?
It's not clear, but I'm assuming we're talking repliQa.
On repliQa. Yes, absolutely. I mean, people used repliQa to detect SNP using Sanger sequencing or other next generation sequencing method. It worked really well. Right. And you can also see from the data coming from the Sanger Institute showing that the single nucleotide detection efficiency of repliQa is comparable to all other high fidelity master mixes tested in the reaction. So yes.
If there's any other questions, please do submit them. I guess I can also let you know for those who aren't aware, but we do have free of charge samples of repliQa and UltraFlex kit and those for those who want to give it a try. So that's something to please consider. But if not, we'll stay on for a couple more minutes just in case somebody thinks of a last minute question. But for those of you who need to leave, I want to thank you very much for your time, for joining us. I hope this was of interest to you. There's additional information on our website, and please reach out with any questions that you may have. Thank you, Subrata. It was a great webinar. And hope the rest of you have a pleasant rest of your day and the rest of your week.